welcome to PW English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you to this lecture in the UMEED NEET English batch. In today's lecture, we are going to be discussing the chapter Microbes in Human Welfare in one shot as always. So, as the name of the chapter itself indicates, we are going to talk about how microbes are useful in our daily lives. What are the different ways in which we make use of them? What are the various products that we obtain from them? How exactly are they useful? And all about that, alright? Before we start discussing the concepts, as always, I'll take you through how many questions have been asked from this chapter over the last few years, starting from 2016. There is just 1 year, 2023, where not even one question was asked from this chapter, but otherwise you can see there's a range between 1 to 4. On an average, you can say about 2 questions. That's just how much you can expect in NEET 2025 as well. This is a very simple, very short, easy to understand chapter, but there's a lot to remember. So this chapter requires frequent revision because there's a lot of names of microbes, what are its products, what are its uses. It's a lot to remember, so it requires frequent revision. And whether you should refer the old NCERT textbook or new NCERT for this chapter, you could refer either of them. Both of them will work. Okay? Now, what are microbes, the heroes of this chapter? What are microbes? Microbes is short form for microorganisms. What does micro mean? As biology students, I'm sure you all know what micro means by now. Micro is something that's super tiny. How tiny are these microbes? They are too tiny to be visible to your naked eye. You cannot take a look at them unless you're looking under a microscope. They're like super tiny, not visible to your naked eye, only visible under the microscope. Such living organisms are called microorganisms. In just general layman English, you call them germs, right? Now, where do we find these microbes? Microbes are present everywhere around us. They are present on the land that we live on. They are present in water bodies. They are present in the air that we breathe. They are present on the surface of our body. They are present right within our body. They are pretty much everywhere. And there are certain species of microbes that are found even in the extreme climatic conditions. For example, you can find certain species of microbes that thrive and grow really well in extreme cold, like even under several feet of snow, you can find them growing. Some of them are the opposite extreme. They grow really well at temperatures as high as 80 to 100 degrees Celsius, where our entire existence could melt off, like our bones could melt, right? So at such temperatures, microbes, certain species of microbes are known to thrive. And also you can see them in hydrothermal vents where the temperature is very high. I think I just mentioned that. And some of them cause diseases. About the microbes which cause diseases, you've learned in the chapter Human Health and Disease. So in this chapter, we're going to talk about the good side of microbes and how in various ways they are beneficial to us in our daily lives. All right? Yeah. So some of them, like I mentioned, they live in extreme hot temperatures as high as 100 degrees Celsius. They are found deep in the soil where there is not sufficient oxygen. They will grow under several layers of snow where the temperature is very cold, wherever the temperature is highly acidic. I'm not sure if you remember in the chapter biological classification, we discussed a group of monarans called archaebacteria. I told you how these archaebacteria live in extreme conditions, extreme weather conditions. I told you there is thermoacidophiles that prefer to live in hot temperatures, highly acidic, low pH conditions. So all of these are microbes, all right? Now, what are the organisms that are grouped under microbes? Like whenever you have to group anything, there is a certain criteria that we have to follow. Like for you to call an organism as a plant, it should have chlorophyll, it should be able to photosynthesize, and it should have a cell wall around the cell membrane. If it has all of that, you can say it is a plant. If you want to call something an animal, you know it lacks a cell wall, you know that it does not have plastids, you know that it has centrioles. So these are things that distinguish plants from animals. So whenever you're checking those criteria, if certain criteria are fulfilled, you can say that organism belongs to that particular group. Now over here, when we classifying microbes, there is just one criterion we're looking at. Is that visible to your naked eye? Is it not visible? If it's visible, then it does not belong to a microbe. If it is not visible, then it for sure will belong to the microorganisms, right? So it will be grouped under microorganisms. Which are the organisms that you can't see with your naked eye? Organisms that are unicellular. Which are these unicellular organisms? They are your monerans and protists, and of course your acellular organisms like viruses, prions, viroids, all of these. Things that are too tiny to be visible to your naked eye, they are called microbes, right? So it includes bacteria, it includes protozoa, fungi, some of them, not all of them. If the fungi are visible to your naked eye, they are not microbes. The unicellular fungi like yeast, they are microbes, alright? Viruses, viroids, prions, all of them are grouped under microorganisms. Because we are not using many criteria for the classification of microbes, the only criterion we are using is whether they are visible to your naked eye or not. It of course will be diverse, right? Diverse means variety. Showing variety. Because there is not much criteria we are using for classification of microbes. Of course, as long as they are very small to be visible to your naked eye, they will be grouped under the same category microbes, and they are bound to be very different from each other. So you do see a high degree of variety or diversity in the microbes. Alright, now how do we study them? They are not visible to the naked eye. How does one even study them? The study of the study of microbes is called microbiology, similar to how you have genetics, physiology, cytology, histology. Even microbiology is a branch of biology where we study about microorganisms. If they're not visible to your naked eye, how does one even study them? Of course, the answer is very clear. It's very obvious. You look at them under the microscopes and study them. But if you have to grow them in laboratory conditions, there are certain steps that we follow. Okay, we maintain sterile conditions. If you're growing certain microbes that I'm studying in, I'm interested in, let's say, one species of bacterium, I'm studying them, I'm growing them in my lab. Because I'm growing this bacterium in my lab, it becomes my responsibility to provide it with food, right? I want my bacteria to grow well so that I can study them better. So in order to make my bacteria grow well, it could be any species or any microbe, but I'm just giving you bacteria as an example. So if I have to study them, I want them to grow well. So I will give it like the fanciest resort treatment. Like when you go to resorts, what do you get? You wake up and you have your meals ready, you don't have to cook, and there's a pool, and you can swim whenever you relax, whenever you feel like, and there is AC in every room that you can control the temperature, whichever is convenient to you. So like that, even in the lab when I'm growing microbes, I will give all of the conditions that it desires to grow well, that it requires to grow well, right? So I will grow it in a temperature which is most optimum for its maximal growth. I will provide a jelly-like nutrient medium which has all of the nutrients that the microbes require to grow. And I will keep it in conditions where no other microbe can enter. I will maintain sterile conditions and I will grow them. Of course, one single bacterium I will not be able to see with my naked eye. But let's say I have a nutrient medium in the form of a plate, like within the plate I have a nutrient medium. In that nutrient medium, I would have added a nitrogen source, a phosphorus source, a sulfur source, whatever else the bacterium requires to grow, everything else, all the food nutrients I've provided in that medium. So I've added agar to it, so it's become jelly-like. Alright, on this I'm going to spread out my bacteria, place it in temperature which is optimum for its growth and allow it to grow. Initially, of course, I will not be able to see anything there because bacteria are super tiny. They measure only in microns, a few microns in length and diameter. So I will not be able to see anything on it. But what happens eventually is, if this is my Petri plate and I've plated my bacteria over it, I've spread out the bacteria over it, initially I will not be able to see anything. But one bacterium, by utilizing the nutrients present in the medium, will divide, grow into 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and so on. It will start growing, and after a few hours or a couple of days, you can see how each bacterium would have synthesized its own colony. Now these colonies are not microscopic. Colonies are easily visible to your naked eye. Each colony will have thousands of individual microbial cells. Alright, so when we study microbiology, we study about individual cell structure and all of that, and we also study about the structure of the colonies. So this is how a bacterial colony looks like, and here is how a fungal colony looks like. So when we study these also, when we study microbiology. Now, like I mentioned earlier, general thought or belief about microbes is that they are very bad for you. Germs are bad, they cause diseases and all of that, but not really. All microbes are not bad. Just like how you have good humans, bad humans, there are good microbes and bad microbes as well. Just one moment. Yeah. So the bad microbes are known to cause diseases. The good ones do us good. Okay. What are the different ways in which it helps us? We will learn about as we move on in this chapter. Okay. Yeah. So first we will learn about how microbes are useful in our daily life, in our household products, starting with curd. I cannot imagine my life, my diet without curd or idli or dosa. Idli, dosa and curd, all of these are microbial products, okay? You know what happens? How curd is formed? It is formed because of a group of microbes called lactic acid bacteria. In short, we call them LAB. One of the genera of lactic acid bacteria is called lactobacillus. Many such genera and many such species are there. Collectively, we call them lactic acid bacteria. So, what do these lactic acid bacteria do? They undergo lactic acid fermentation. What they do is, they are naturally present in air also. They will convert milk into curd. How exactly does this conversion happen? Milk contains certain proteins, certain sugars, a little bit of fat and all of that. There is proteins in the milk that are called casein. The name of a protein in milk that is found in milk is called casein. These lactic acid bacteria act upon that casein and partially break it down and produce lactic acid as a product, and in this process milk will transform into curd. Okay, so in curd you can see millions of lactic acid bacteria present. Okay, so they grow in milk and convert it into curd. During the growth, lactic acid bacteria produce acids that coagulate and partially digest the milk proteins. Okay, small amount of curd. Okay, you all must be aware that sometimes when there is not enough curd at home, your mother or your father or whoever does this at home, they might ask you to go to your neighbor's house, get like a little bit of milk, a little bit of curd, so that they can use it for preparing curd in your house when you've run out of curd. So we call that inoculum. Inoculum, in Kannada we call it heppu. I don't know what it's called in other languages. So you take a little bit of curd, like just 1 spoon is sufficient. Just warm, heat the milk just a little bit, make it a little warm and add this 1 spoon of curd to it. If you leave it for a couple of hours, whatever bacteria were present in curd, they will start acting upon the milk proteins, break it down partially and convert that also into curd. So that little bit of curd that we add is called the inoculum. Okay, even that 1 tablespoon of curd will have thousands and millions of lactic acid bacteria. Now, why is curd important? Curd is important because of 2 reasons. The first reason is it's nutritionally better than milk in terms of its vitamin B content. Milk also probably has a small amount of vitamin B, but curd has even more compared to milk, even more quantity of vitamin B. Secondly, you know that we're constantly eating outside food. Sometimes you're eating street food that may be contaminated. You never know, but you eat that, it goes down your digestive tract. These good bacteria that are there in the curd, I'm sure you've heard that curd has probiotics, right? When we say it is probiotic, it means it has microbes, okay? So what these microbes do is, they are like your good gut bacteria. If by accident you consume some food that is contaminated with a microbe that could potentially cause any disease in your body, this will not allow that microbe to grow. So potentially pathogenic microbes, if by chance they enter your digestive tract, the good gut bacteria that are present probably in your intestine, they will not allow it to grow and spread in your body, and thus it protects you from diseases. Okay? So it improves the nutritional quality by increasing vitamin B12, and it also helps in checking disease causing microbes. That's about curd. Next, I told you, I'm a South Indian. Idli and dosa are staple food in my household. Every week I make idli dosa, and fermented good is good for your gut because again it has microbes, so it will take care of whatever bad bacteria enters your body. Okay. You may have noticed when your mother prepares batter for dosa or idli, she leaves it for a couple of hours, usually overnight. We grind the batter for dosa idli around the evening and you leave it overnight. If you notice, the batter would have risen up. Okay? So it would have become slightly spongy in appearance, slightly porous, and it would have risen up. Now, the reason why the batter rises is because of fermentation. Who is performing this fermentation inside this batter? It is bacteria. Over here, which species of bacteria is not mentioned, so you don't worry about it. Where is this found? Generally, it will be found on the surface of your rice grains and your dal, the same bacteria. Whenever you're making dosa idli batter, we use soaked rice and dal, right? On its surface only these bacteria will be present. Once you grind it and keep it overnight or for a couple of hours, they will start undergoing fermentation over there and release carbon dioxide, as a result of which it will get its puffed up appearance. Okay. So over here the dough used for dosa and idli are fermented by using bacteria. Yeah. And the puffed up appearance of dough is due to the production of carbon dioxide gas. When their fermentation is going on, carbon dioxide is released as a byproduct. So that is what gives rise to that puffed up appearance. Okay, that's about dosa and idli. Next, cakes, bread, your pastries, all of that in the baking industry, microbes are used extensively. Specifically, a unicellular fungus or yeast called Saccharomyces cerevisiae is used in this process. Because of its extensive use in the baking industry, it's also called baker's yeast. Okay, so whenever you bake a cake or some bread, you know that even over there you will have this spongy appearance. That is because of carbon dioxide being released as it gets heated. As it's getting baked, whatever carbon dioxide is present, that will get released, cause that porous appearance in bread and cakes. Okay, you cannot even imagine having like cutting birthday cakes during your birthday parties. If not for microbes, there wouldn't be cakes, there wouldn't be a baking industry, right? So they are important. And then in South India, we have a traditional mildly alcoholic drink that we obtain from the sap of palm trees. You know palm trees, right? They will cut, one person will climb high on top of the palm tree, they will cut a slit at the top, and then the sap of the palm tree will start oozing out. They will tie a pot around the tip of the tree for the sap to collect, and they will allow that to undergo fermentation. After a couple of hours, you can see that it will become mildly alcoholic and that is consumed. Okay, and that is called toddy in English. It's called kallu in Kannada. I don't know what it's called in the other languages. So yeah, this drink that is mostly consumed near the rural side of South India, that also fermentation happens because of microbes itself. Next, this is not very common in India, but you can find it in other Asian countries where they ferment fish, they ferment bamboo shoots, they ferment soy. All of this is fermented and consumed as a delicacy. Even over there, if fermentation is happening, that is because of microbes. All right. Now, talking about cheese. Cheese is a milk product. All types of cheese is produced from milk itself. You may have heard of different types of cheese. There's Swiss cheese, there's Roquefort cheese, there's cheddar cheese, there's mozzarella cheese. There are different types of cheese. What differs from one cheese to another? Two things change. The source of milk. Generally, most cheese is made from cow's milk. Sometimes it could be sheep or goat milk also. So the source of milk varies. And which is the organism that is used to ferment the milk, that also varies. Because of these two differences, the texture, the smell, the flavor, and how the cheese looks, all of this will vary. All of them ultimately are milk products, but milk of which animal varies, and which is the organism that is fermenting the milk to form cheese, that also varies. Because of that, you get different types and varieties in the texture, flavor, taste, and fragrance of the different types of cheese. In your syllabus, there's a mention of two different types of cheese. First one is your Swiss cheese and the second one is your Roquefort cheese. Swiss cheese, I'm sure everybody must have watched this in Tom and Jerry cartoon where Tom is always trying to trap Jerry using this cheese as the bait. So it has like really large holes in them. This cheese is produced by a species of bacterium called Propionibacterium shermani. And even over here, the reason why these holes are formed in cheese is because of carbon dioxide. During the process of fermentation, huge amounts of carbon dioxide gets released and that will cause the formation of holes. It's not like somebody has got like a punching machine and they've punched holes out of this cheese. It is formed, the holes, the large holes are formed because of carbon dioxide. Remember, Propionibacterium shermani is the bacterium used for the formation of Swiss cheese. Talking about Roquefort cheese. Roquefort cheese is very popular in Peppa Pig. If you watch Peppa Pig, you know Daddy Pig loves one kind of cheese that has like blue stripes on it. It has like a weird odor that Daddy Pig absolutely loves. It is this Roquefort cheese. Now this cheese is ripened by using a species of fungus they've mentioned in the textbook. They've not really mentioned the name of the fungus. It is Penicillium roqueforti, but they've not mentioned it. So just remember, Roquefort cheese is ripened by growing a specific fungus on top of it, which gives it a particular flavor and odor also. Alright, so that's about cheese. Now, we've completed all of the points that was given under use of microbes in household products, right? We started off with curd, then we did dosa idli, and then we spoke about how it's used in the baking industry, then we spoke about how it's used in toddy making, and also about how it is used in the formation of different types of cheese. Now let's dive into the use of microbes in industries at the industrial level. There are very many industries that rely entirely on microbes. If not for microbes, those industries wouldn't even exist today. Alright, so in huge industries, like if you're doing it at a small scale for your household, you will have like 1 spoon of inoculum, enough to make curd for a family of 5 people. And probably a few liters of dosa or idli batter will suffice. So you can do it at a small scale. But if you're doing it at an industrial scale, you need to maintain, sterile conditions for those microbes, give it that resort kind of treatment at a larger scale. So you need instruments and equipment that can handle that kind of quantity, right? So generally when microbes are grown at industrial scale, there are fermenters used, okay? Fermenters are basically very, very huge vessels in which sterile conditions are maintained. Depending upon which microbe you're growing in these fermenters, you can alter the temperature, you can change the nutrient medium after they are getting used up. You will have an outlet to collect the product of the microbe. All of those provisions will be provided. Okay. So example for microbes used in industrial scale is alcoholic beverages and also antibiotics. All right. Now let's talk about alcoholic beverages. The microbe that is used in the production of alcohol is Saccharomyces cerevisiae. Remember, we've learnt the same thing in baking industry also. I told you it's called baker's yeast. Since it's extensively used in the brewing industry for the production of alcoholic beverages, it's also called brewer's yeast. Okay? It is used in the production of beverages like wine, beer, whiskey, brandy or rum. Remember, I told you how there are many different types of cheese and one cheese differs from another cheese because of the source of the milk probably, or because of the organism that is used to ferment it. Here you have different types of alcoholic drinks. All of these drinks are fermented or formed from Saccharomyces cerevisiae itself. But why each of them has different taste, different texture, different flavor is because of what is used as the substrate. Okay, if like fruits like grapes are used, then you can make wine out of it. If barley is used, I think from barley they make beer. If I don't remember very clearly, potatoes are used, I think tequila or vodka or some alcoholic drink of that kind is made. I'm not very sure because I don't drink myself. It's something that I've read. Okay, so it's not very strongly stored in my memory. So depends on what substrate the organism acts upon. So yeah, that's about it. And you will see that in these different alcoholic drinks, the amount of alcohol also varies. How much content of alcohol is present is also varies. In some of them it's less than 10%, and some of them it could be as high as 15, 20, 30% of alcohol also present. Okay. Now, if you remember, when we discussed the chapter respiration in plants in the UMEED batch, I taught it myself. I told you how whenever yeast undergoes fermentation, alcoholic fermentation, it will start producing alcohol. Now imagine there is yeast growing. There is a substrate of malted barley given to it. It is acting upon this barley. It is producing alcohol as a byproduct. It's growing in the same medium. So as this fermentation proceeds, alcohol content keeps on getting increased in the medium. At one point when the alcohol reaches about 12 to 13%, the yeast itself will find it difficult to grow in such a medium. The alcohol content will start becoming toxic for that yeast and it will start dying. Okay. Now, if the concentration of alcohol cannot exceed 13% because yeast themselves are dying there, how is it possible that alcoholic drinks contain higher than 13% alcohol? I told you how some drinks like whiskey can contain as high as 20% alcohol also. How is that possible? That is possible because of a process called fractional distillation. You may have learnt about fractional distillation in physics or chemistry, probably about petroleum. When you learn, you get a lot of byproducts from petroleum like Vaseline and a lot of things. Because you're heating at a particular temperature, it will evaporate at different components in that mixture will have different boiling points. According to its boiling point, it will start evaporating and you will start condensing it and collecting it at different intervals. When that kind of fractional distillation is done for this alcohol, since alcohol has a much lower boiling point compared to water, it will evaporate first, and then after that fractional distillation you will get a higher concentration of alcohol in certain alcoholic drinks. Alright, over here it's important for you to remember which ones are distilled alcoholic drinks and which ones aren't. Among the given 5 examples, the ones that are obtained without distillation are beer and wine. The ones that are obtained after the process of distillation is whiskey, brandy and rum. Okay, remember this will have a higher content of alcohol compared to this. Okay, now that's about microbes used in brewing industry. Now let's talk about the use of microbes in the production of antibiotics. Now what are antibiotics? I'm sure you've heard about antibiotics. Antibiotics are drugs, medicine that is routinely prescribed by doctors to treat any bacterial infections. If you have any bacterial infection, I'm sure you would know that doctors will prescribe you antibiotics like penicillin, amoxicillin and those kind of things. Now, what does the word antibiotic mean? Anti means you know, right? Anti means against. Bios means life. What are these antibiotics? Why are they even produced? Now let's say there is a medium, okay? So let me just draw something random here. This is some substratum. On this, 2 different groups of microbes are growing, okay? One group of microbe, one species of microbe, I will represent in this bright green color. And utilizing the nutrients from the same substratum, a contrast blue color, 2 species of microbes are growing. Now there is competition between these 2 species. What are they competing for? They are growing on the same area, so there's competition for space. They are utilizing the nutrients from the same substratum, so there is competition for nutrients. Right? So what happens here is, in some microbes over the course of evolution, they have attained the capacity to produce chemicals that can prevent the growth of other microbes. These chemicals won't harm the microbes that are producing them. These chemicals will specifically target bacteria and in one way or the other prevent them from growing and increasing in number. How will that help the microbe that is producing them? Competition is less. If the number of this type of bacteria reduces, then all of the space is for this type of species and all of the food and nutrition is available for this. So, in order to reduce competition, this is one of the mechanisms that certain microbes have evolved over the course of evolution. Now, what happens is, let's assume the one that I've drawn in green color, that is one type of microbe, microbe 1. This has the ability to produce certain chemicals, and those chemicals are called antibiotics. Now, whenever this blue color microbe is growing, increasing in number, it comes in contact with antibiotic, it will stop growing. Either its cell wall formation doesn't happen or DNA replication is inhibited. By one way or the other, this chemical has the ability to slow down or retard or completely stop the growth of the other microbe. That is the reason why we are calling it antibiotic. Anti means against. Biotic means, bios means life. So this chemical is against life for this particular microbe. Antibiotics against life, if you eat that, it's against life for you. It is not like that. It is against life for those bacteria which are susceptible to it. Okay? So that is antibiotics. Understood? We got to know this from Alexander Fleming. He is the person to have first discovered antibiotics, and ever since then it has been utilized extensively for the treatment of bacterial diseases. I want you to remember how these antibiotics do not treat viral infections. They are ineffective against viral infections. They specifically treat bacterial infections. Sometimes when you have like a runny nose and a sore throat and you go to the doctor, they will first give you certain medicine. They will immediately not prescribe antibiotics. They will tell you if it still remains after like 2-3 days, come back. And then when you go back, it is confirmed that it's a bacterial infection, only then will they prescribe your antibiotics, right? Because antibiotics taken for viral infections is of no use. All right. So before the discovery of antibiotics, many large number of people used to die for diseases like plague and whooping cough and leprosy and diphtheria and all of that. But after the discovery of antibiotics, all of these diseases have now become curable. And let's talk about this interesting story of how Alexander Fleming happened to discover antibiotics. He was not looking to discover any chemicals that had antibiotic properties. It was a chance discovery. He was working on something else and he accidentally stumbled upon this discovery. So he, in his lab, was working on a species of bacterium or a genus of bacterium called Staphylococcus. Okay, so he was growing it on he was working on Staphylococcus bacteria. I told you initially how when we're growing microbes in the lab, we give it food. We have to maintain sterile conditions. When I say we have to maintain sterile conditions, we have to clean everything with alcohol and make sure, except for the bacterium that you're interested in and growing, you do not want any other microbe to enter. You have to maintain that because you're spending energy and money in preparing the nutrient medium. You do not want some random microbe to come and utilize it and grow and it is of no use to you, right? So you have to maintain sterile conditions. So when we try to maintain sterile conditions, there's a lot of things that we do. We treat the area that we're working with UV. We clean everything with alcohol. We wash everything with alcohol. So apparently, while he was working with Staphylococcus bacteria, he had not cleaned one of his plates properly. He had not washed it properly, which means he had not cleaned it correctly with alcohol, and it ended up becoming contaminated with a species of mold, a type of fungus called Penicillium notatum. What he noticed was, I want you to look here, everything that I'm marking in this green color was the Staphylococcus that was spread out on the plate for it to grow. But in one location on the plate, accidentally, because he had not cleaned and washed his plates properly, he ended up having it contaminated with Penicillium notatum. Okay, so this is Penicillium right here. And around it what you have is your Staphylococcus. Alright, I want you to notice how wherever penicillium is growing, immediately around it, Staphylococcus is unable to grow. Why is the Staphylococcus unable to grow? Because this penicillium has released certain chemicals that are not allowing the Staphylococcus to grow. And then he isolates that chemical and he calls this chemical penicillin because it's obtained from Penicillium fungus. Generally, antibiotics are obtained from bacteria as well as fungi. Both of them produce chemicals, these chemicals for its defense and to reduce competition, but it is effective against bacterial infections only. Okay, so this is a fungus that produces it, but it's effective, it kills bacteria. Okay, now he is the first person to have discovered it, but he did not use it to its complete potential. Two other people, okay, Ernest Chain and Howard Florey, they are the ones to have utilized these antibiotics to its maximum potential, and they used these chemicals to treat wounded soldiers, American soldiers in World War II. You know, when they become wounded, when there is an open wound, it is highly, vulnerable to get infected. Bacteria can easily enter the body through those wounds. So they used these antibiotics discovered by Alexander Fleming to treat wounded soldiers, and as a result, for this significant, very, very significant discovery, I'm sure you understand how significant it is, because before the discovery of antibiotics, leprosy patients used to be outcasted. They used to be sent outside of the villages for them to live alone, and then thousands and millions of people died because of plague. All of these could now be easily curable. So it is a huge milestone in the field of medicine, and truly deserving. All three of them were awarded with the Nobel Prize in the year 1945. Okay, that's about antibiotics. Now let's talk about certain acids, chemicals that we obtain from microbes. There are 4 acids that you should remember: A, B, C, L. Acetic acid A, butyric acid B, citric acid C, lactic acid L. Lactic acid, you already know lactic acid bacteria, you already know lactobacillus, so that is already done. Which is this ABC? Acetic acid is obtained from a bacterium called Acetobacter aceti. It's in the name itself. Butyric acid is obtained from a bacterium called Clostridium butylicum. Citric acid C is obtained from a fungus called Aspergillus niger. These are things you should remember. Alright. Apart from this, we also obtain certain enzymes from microorganisms. First one is lipases. You've heard about lipases in digestion and absorption. It is one of the enzymes that are produced by our digestive system. Also, some amount of salivary lipase also is produced. Lingual lipase is also produced in your mouth that will break down lipids. So microbes also produce these lipases. Lipases are basically enzymes, hydrolytic enzymes that act on lipids and break lipids down. Okay. So where could this be used? It is used in detergent formulations. Whenever you have oily stains on top of your laundry, on top of your clothes, to remove the oily stain, lipases are used. Detergents are not made up of lipases only. In the detergents, you will have different types of enzymes and different other chemicals as well. But along with that, lipases also will be present, and that is what will break down the lipids or oily stains that are present on top of your clothes. So lipases are used in detergent formulations, and it removes oily stains from the laundry. Second, pectinases and proteases. What is pectinase? Pectinase is an enzyme that breaks down pectin. What are proteases? Proteases are enzymes that break down proteins. Where do we find pectin? Pectin is found in the cell wall of plant cells. Proteins are also found in plants. What is the use case of proteases and pectinases? These are used as clarifying agents or clearing agents whenever bottled juices are manufactured in industries. You make orange juice at home, buy like a Minute Maid or some other Maaza or whichever orange juice from the market, bottled juice. Look at their appearance. The ones that we make at home will have like a hazy appearance, and the ones that we get from the market bottled juices, they will have a clearer appearance. The reason why we have a hazy appearance is when you grind it in the mixer, pectin will remain, some amount of pectin will remain as it is and some proteins will remain as it is, that will give it a hazy appearance. When they add proteases and pectinases to boiled juices, whatever is causing that hazy appearance will get broken down and it will get like a clearer appearance. Alright, so they are used for clarifying bottled juices. Next, streptokinase is an enzyme, very important. It has medicinal use. It is obtained from a bacterium called Streptococcus. Now what does this streptokinase do? Streptokinase is also called the clot buster. You can guess from the name what it does, right? It removes clots from your blood vessels. And generally the streptococcus is modified genetically and then streptokinase is obtained from it. You know, in our blood vessels, sometimes it is possible for clots to form. If there is a blood clot in the blood vessel that is supplying blood to your heart, you know, heart is the one that supplies blood to all parts of a body. For heart to function normally, it requires oxygen to beat, contract, it requires nutrients to contract. So it requires its own blood supply. So whenever the blood supply to heart is kind of blocked because of a clot, heart may not receive sufficient amount of nutrients and oxygen, and that is called myocardial infarction, as a result of which heart attack can happen. So in such cases, to remove the clots in the blood vessels, streptokinase is used as a clot buster. Alright, so it is modified by genetic engineering. I mentioned that it removes clot from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. That is why we call it the clot buster. Now let's talk about some other bioactive molecules. There's two more of them, cyclosporin A and statins. Cyclosporin A is an immunosuppressive drug that is obtained from a fungus called Trichoderma polysporum. Cyclosporin polysporum. Okay, remember it that way. So what is an immunosuppressive drug or an immunosuppressive agent? As the name itself indicates, it suppresses the functioning of your immune system. What is the job of your immune system? The job of your immune system is to protect you, right? Protect you from what? Protect you from anything foreign. It could be something as simple as a pollen grain. It could be some potentially, a potential pathogen that can cause disease in you. Your immune system knows what belongs to you and what doesn't. Every time it encounters something that is not part of your body, it will fight against it so that you are safe, you do not get any infections. So whenever people have undergone transplants, organ transplants like a liver transplant, kidney transplant, bone marrow transplant, our immune system recognizes the transplanted tissue as a foreign tissue. It is very quick in recognizing what does not belong to your body. It could be from a donor who's like close family, there is a match and everything, but still it recognizes what does not belong to you. And let's say that person's both of the kidneys had failed, and if he has to survive, if he has to live, he needs a kidney transplant. He's got kidney transplant done, but now his own body's immune system will recognize that kidney as foreign and start to destroy it. You do not want that to happen. So you will provide this person or the patient who's underwent transplantation of any organ or tissue with immunosuppressive drugs so that the immune system kind of calms down and doesn't really affect the organ that has been transplanted. Now there is a downside to this. If a person is on immunosuppressive drug, let's say that person who had got kidney transplant is on immunosuppressive drug, so his immune system is not recognizing the kidney as foreign. But if he has been exposed to some other pathogen because of the air that he breathes or because of some food that he has eaten, there is some other pathogen that has entered in his body which can cause disease, immune system fails to identify that also. It's not like don't identify the kidney but identify one bacterium that can cause disease. It doesn't work like that. If your immune system is suppressed, it cannot identify pathogens, potential pathogens. It won't even identify the transplanted tissue or organ as foreign. So such people should avoid going in crowds, meeting a lot of people and be isolated, and they will be on immunosuppressive drugs for the rest of their lives. Okay, so it has a downside, and the the positive is that they will have a longer lifespan because of the kidney transplant or whatever organ transplant. Okay, next one, statins. Statins are also called blood thinners. You may have seen elders in your family taking that, your grandmother, grandfather, anybody. And this is obtained from a fungus, a species of yeast called Monascus purpureus. Now, what does this do? This helps in lowering the cholesterol level in the blood. And how exactly does that do it? For any chemical reaction to take place in our body, enzymes are required. There is an enzyme that is required for the synthesis of cholesterol. Okay, so let's say this is the substrate. When this substrate goes and binds to this, the substrate will get converted into the product, which is cholesterol. Now, these statins resemble the structure of this substrate. So, they will compete with the substrate for this active site. If it goes and binds here, the substrate will not be able to bind there. If the substrate doesn't bind there, cholesterol will not be synthesized. So, cholesterol synthesis will come down. Alright. So, by competitively inhibiting, it will reduce the synthesis of cholesterol and thereby brings the level of cholesterol down in blood. Okay. Now we have to talk about how microbes are used in sewage treatment. So first, what does sewage mean? Sewage basically refers to the municipal waste that is generated in the cities, municipal wastewater that gets generated in large amounts on a daily basis in cities and towns. It includes your kitchen waste, it includes the water that gets out of your washrooms and bathrooms. So this sewage or municipal waste will contain high quantity of human excreta, a lot of microbes. Some of them could be potentially pathogenic. And since human excreta is a main part of it, and it includes some amount of kitchen waste as well, it will contain high quantities of organic matter. Now, ultimately, all of the wastewater that gets out of our households and town, cities, houses, and these industries and hospitals, all of them ultimately will be passed on to water bodies like rivers. But since this municipal wastewater could have pathogenic microbes, if there are humans that are consuming water from the nearby river, they could get diseases. Since there is high amount of organic matter, it can cause eutrophication of the water body. It can cause pollution in the water body. So before we introduce or pass on this sewage into water bodies, it has to be treated. So it is sent to sewage treatment plants where organic matter gets reduced and then it is passed on to water bodies. Instead of directly passing this on to the water bodies, which will cause pollution, it is treated first and then passed on. When it is treated, what exactly is happening? This organic matter itself will cause pollution. So if you do not want this water sample to cause pollution in the water bodies, you have to make sure that the organic matter content has come down. How exactly that is done, we will learn in sewage treatment. Alright. So in sewage treatment, basically two stages, it happens in two stages. The first stage is called primary treatment. In primary treatment, there are two steps. In the primary treatment, microbes are not utilized. Keep that in mind. It's a physical process. Firstly, something called sequential filtration takes place. Sequential filtration. You know, sewage will have a lot of debris or waste that floats on the surface of the water sample. So this sequential filtration removes all the floating debris. Sequential filtration means they will pass on. There will be tanks present, and in these tanks there will be filters arranged in a sequence of varying pore sizes. For example, first there will be a filter that has this much pore size. Right under that there will be a filter with a smaller pore size. Under that there will be a filter with an even smaller pore size. Under that there will be a filter possibly with the smallest pore size. Now you're adding your sewage into this. When you add your sewage into this, in the sewage, whatever floating waste or debris is present, that will get filtered out. So let's say within the sample of sewage, there was a chocolate wrapper. It can easily pass through this. It cannot pass through this anymore, so the wrapper will get filtered out here. Let's say there was a water bottle. That will get filtered out here. So like that, in different levels, in different filters, whatever floating waste is there, that will get separated out. What they do next is called sedimentation. During sedimentation, the denser particles like soil and pebbles, these are removed. So they will allow this filtered sewage to pass on to this tank. In this tank, what they basically do is, they will allow the sewage to stand undisturbed for some time. Whatever heavier, denser particles are present in the sewage water, in the form of soil and pebbles and all of that, that will start settling down at the bottom of the tank. This tank is called a settling tank, or let's just call it primary settling tank, because it's part of the primary treatment. If you allow it to stand still for a while, it will all settle down and form a sediment at the bottom. Right now, this is called the primary sludge, and whatever effluent remains on top, this is called the primary effluent. That's all is done in the primary treatment. Now, in the secondary treatment, I'll tell you what happens. Let's do it in the next slide. Okay. Yeah. So next, this primary effluent right here, this primary effluent, well, let me write it on the other side. Primary effluent will be passed on for secondary treatment. Now, what exactly happens during secondary treatment? Let's understand that. The primary effluent will be passed on to a tank that is called aeration tank. Into this aeration tank, we will pass on the primary effluent. Now, what we know is, in this primary effluent, there will be large amounts of organic matter. Large amounts of organic matter is there. Okay, let me just label this as organic matter. Along with that, there will be a lot of heterotrophic microbes as well, which is fungi and bacteria. I will represent fungi like this in the form of filaments, thread-like, for our understanding. Of course, it won't be pink in color. And let me use orange to represent bacteria. All right, this is the bacteria. These are naturally present in sewage. They are heterotrophic in nature. The whole idea why we are treating the sewage is to ensure that the organic matter content comes down. To make sure the organic matter content is reduced, we are going to make use of these microbes. Now, these fungi and bacteria that are there in the sewage will make use of oxygen and break down organic matter. They use organic matter as food. They oxidize the organic matter. From that they get energy. Utilizing the energy, they will increase in number. So we are making sure that the fungi and bacteria present naturally in sewage utilize the organic matter as food. For that process, oxygen is required. To ensure that they get sufficient oxygen for breaking down the organic matter and reducing the organic content, there will also be stirrers present inside, which will ensure circulation of the fluid and ensure uniform availability of oxygen and air inside this water sample. As it keeps spinning, oxygen is made available. Eventually what happens is, these bacteria and fungi will make use of organic matter. As they are utilizing organic matter as food, organic matter content will keep coming down. As they are eating that food and growing in number, consuming that food and growing in number, the number of bacteria and fungi will start increasing. Now, how does one know whether the organic matter in a water sample has decreased? Is it decreased significantly? Is the organic matter content very low now that I can, so that I can now let it into water bodies? If you have to check it, there is one way you can check it. That is in terms of what we call biochemical oxygen demand. Biochemical oxygen demand. In short, this is abbreviated as BOD. Now, let's say this is like a water sample that we have taken previously. Okay, I have taken like 1 liter of that water sample. I have taken 1 liter of that water sample. In this water sample, there is some amount of organic matter and there is some amount of, I think I used orange and pink. Yeah. Oops. Bacteria, and there is some fungi as well, which are heterotrophic. They are using them as food. Right now, what is the demand of oxygen here? In this 1 liter of water, whatever organic matter is there, to oxidize that organic matter, how much oxygen do the microbes need? The microbes are oxidizing organic matter. For that, oxygen is required. In this 1 liter sample, how much ever organic matter is present, in order to break it down and oxidize it, how much oxygen do the microbes need? That gives us the biochemical oxygen demand. If there is more organic matter, then the BOD also will be more. the demand for oxygen also will be more. If there is more organic matter, the polluting potential is also high. So whenever the BOD is high, it means organic matter is more, polluting potential is high, so you not let it into water bodies. You will keep checking the BOD at regular intervals. Once you ensure that the BOD has significantly decreased, we will transfer it into water bodies. But before that, something happens. I mentioned how because of aeration, microbes now have sufficient oxygen and they will start breaking down organic matter. Right? And organic matter content will start reducing and these microbes will start increasing in number. So as this proceeds, you can see how organic matter reduces significantly. But right now, bacteria and fungi have grown so much because of their growth. I think I drew too many. Okay, this is sufficient. Yeah. And they have formed mesh-like structures that float on the surface of this tank. This mesh-like structure is made up of filaments of fungi, and within the filaments of fungi, you can even see bacteria being trapped and present. Now we call this flocks. It is a network of fungal filaments along with bacteria. They've increased so much in number that they formed this mesh-like network-like structure. Now you have to remove this before you pass on the effluent into the water body. So what do? You pass on this effluent into a tank called the settling tank. In the settling tank, what happens is the effluent, of course, will be there. As you allow the effluent to stand still undisturbed for some time, whatever flocks were earlier floating on the surface will now settle at the bottom. Okay, so this is still the aeration tank itself. Now it has been passed on to the settling tank. Over here, the flocks will form a sediment, and we call this sediment as the activated sludge. All right. Now, one small portion of this activated sludge is pumped back into the aeration tank as inoculum. The remaining major portion of the activated sludge is passed on to anaerobic sludge digesters. Over here, certain microbes anaerobically act upon this. This will be used as the substrate. Anaerobic microbes act on it and break it down anaerobically and result in the formation of a mixture of gases like methane, carbon dioxide, hydrogen sulfide that are inflammable and can be used as a fuel. Biogas can be produced. What happens to this effluent on top? This effluent, one moment, I think my son is calling me. Press this. Press this. Yeah. All right. And this effluent on top, which has almost very little organic matter, will be passed on to water bodies. All right. So that is how microbes are used in treating sewage. So that's about sewage treatment. Now let's move on. You know how many of India's rivers are polluted, especially the major important rivers like Ganga, Yamuna, they are polluted. In order to reduce the pollution in these major rivers of India, the Ministry of Environment and Forests have initiated certain plans that are called the Ganga Action Plan and the Yamuna Action Plan, which are meant to reduce the rate of pollution, water pollution in these rivers. So the aim of these projects is to make sure that they build sufficient number of sewage treatment plants so that no sewage enters the water untreated. All right? Yeah. So they have proposed to build a large number of sewage treatment plants. Now let's talk about biogas production. Biogas is also called gobar gas because the substrate that we use mostly for the production of biogas is cow dung. Why cow dung, you might ask? You know, for the production of biogas, we need bacteria, a type of bacteria called methanogenic bacteria or simply methanogens. These bacteria are found in marshy areas. They are found in the gut of herbivorous mammals also. Herbivorous mammals are called ruminants. It includes your sheep, goat, cow, all of these organisms, buffaloes that are herbivorous. In their alimentary canal, they will have a part called the rumen. It resides in the rumen. You know, these herbivorous mammals, they feed on plants, right? And plants have plant cells, and plant cells have cell walls that are made up of cellulose. We do not have any enzyme to digest cellulose. It acts like fiber in us. Even cows themselves cannot digest or break down cellulose. These bacteria that are present in the gut of these ruminants, they will help in the breakdown of cellulose and help in better nutrition for the cows. As they break down cellulose, as a byproduct, they also produce methane, carbon dioxide and other gases which are inflammable and can be used as a source of energy. Because they are present in the gut of these animals, large amounts of these bacteria will be found in the cow dung as well. So when they use cow dung for the production of biogas, it will have methanogenic bacteria in it. All we need to do is give it anaerobic conditions so that it can act upon the substrate and break it down anaerobically to produce this mixture of gases called biogas. How exactly is this done? It is done in a biogas plant. Here's what a biogas plant looks like. You have a digester, which is a concrete, which is generally made up of concrete. It is a tank that is made up of concrete, which is about 10 to 15 feet deep. On top of this concrete digester, you have a floating gas holder. Okay, so this thing on top of it is called the gas holder. It's also called a floating cover. There is one inlet through which we introduce the slurry into the digester. The slurry basically includes farm waste, vegetable waste, cow dung. They make like a slurry out of it, liquid consistency, and feed it into the digester. Since the cow dung will already have methanogenic bacteria, we're providing anaerobic conditions, it will start acting upon it and produce the mixture of gases, carbon dioxide, methane, hydrogen sulfide and all of that. And every time the gases are released, this gas holder will start, rising up, and all of the gases are collected here. There will be an outlet through which the gas can be supplied to nearby households, and over there it is used for cooking and lighting. Whenever the slurry gets used up, it is removed from the digester through another outlet that can later be used as manure in the fields because it is rich in organic matter. Okay, so that's about biogas production. And generally, since cow dung and all of this plant waste, agricultural waste is found in rural areas, mostly biogas plants are also located in rural areas. The technology of biogas plant was developed by, okay, well wait, I think I've mentioned this already. Biogas is a mixture of gases containing methane. It is produced by microbes. It may be used as fuel. It will contain methane, carbon dioxide and hydrogen sulfide. It is produced in the rumen of, where is rumen? Okay, it's here. Rumen of cattle. So it will be present in large quantities in them because it helps in breakdown of cellulose. And cow dung contains rich it's rich in these bacteria, so it can be used in the production of biogas. These bacteria are called methanogenic bacteria or methanogens. One species of it is called methanobacterium. All right, I've told you all about it. I've mentioned this also already. I don't know why this slide was repeated, but okay. Excuse me. Yeah. So the technology of production of biogas in India was developed because of the efforts of two organizations. One is Indian Agricultural Research Institute, abbreviated as IARI. The other one is Khadi and Village Industries Commission, which is abbreviated as KVIC. Both of them contributed to the development of biogas production technology in India. So that's about biogas production. Now let's talk about the almost last part of the chapter, which is microbes used as biocontrol agents. What are biocontrol agents? Whenever living organisms are used for pest control and disease control, we call it biocontrol agents. Generally, what farmers do is, in order to control any pest in their crop field or farm which can reduce the yield, they use chemical fertilizers that are available in the market. Now, there are two problems with using chemicals as pesticides. Firstly, since you're spraying it on the plant, we eat that later, some animals eat it later, it could cause, health problems in us. Health issues can arise in us, in animals that consume it. Secondly, if there is rain, it can get washed off into the soil and cause soil pollution. Also, there's another reason. If you're, spraying pesticides, there could be one insect pest that is causing a reduction in yield in your farm. These chemical pesticides don't specifically target that pest. It will kill every other insect also that is present in your farm, which could be beneficial, which could be a pollinator. So it kills the good insects and the bad insects as well, which may not be a good thing. So in order to prevent all of these from happening, we can make use of living organisms itself as pest control agents or biocontrol agents. How do we do that? Let's say there is a crop that you're growing. Let's say you're growing maize or something, and you have maize, you have a maize plant, there is corn growing. Okay, let's say there is one insect or one pest that feeds on it. Okay, so this is some pest that feeds on it. So this is a pest. In nature, there will be an animal that in turn feeds on this. This pest will have a predator, right? It could be another insect, it could be some other animal, some bird, you never know. This pest will have another predator, right? Instead of spraying chemical pesticides on the field, you introduce this predator in the field so that this will take care of this. Only this pest will be destroyed or its number will be controlled. The other insects in your field will not be harmed. There is no chemical poisoning that happens to the food. Animals and humans who consume that food are safe. Soil pollution will not happen. All of that can be avoided. So in order to incorporate this in farming, a farmer just have the knowledge of what are the pests for the crop he is growing, what are the natural predators for that pest he can find. So if he has that knowledge, biocontrol becomes very easy. There are some examples given to us. Aphids are these small insects. Ladybird beetles, they are known to feed on this. So they can control ladybird beetles. Though this chapter is called Microbes in Human Welfare, we are talking about insects here. Okay, they are also biocontrol agents. You know, mosquitoes can be pests in some crop fields. Dragonflies can be used to control the mosquito pests. In the chapter Biotechnology, I'm sure you've learnt about Bt cotton and Bacillus thuringiensis. That also can be used to get rid of butterfly caterpillars. Generally, the plants that this affects or is a pest for are brassicas and fruit trees. Over there, the leaves are fed on by the butterfly larvae, therefore reducing the yield. What we do there is you get Bacillus thuringiensis spores in the form of dry spores. You mix that with water and you spray it on these vulnerable plants. Whenever the larvae feed on them, there will be toxins present in those spores. They will enter the gut. The gut of insects is alkaline. It will get activated there. It will rupture the gut and the organism will die. So thereby you can control these pests. These genes are introduced into plants itself and made it genetically modified, like you have BT cotton, you have BT brinjal. So that is also made, but more about it you learn in the chapter biotechnology and its applications. For now, just understand that such bacterial spores, when they are sprayed, then it can to an extent control the pest. Next, biocontrol agents don't necessarily control pest, they can control diseases also. Trichoderma is a fungal species about which we've already learnt in another in a part before. I told you how Trichoderma polysporum produces cyclosporin A, which is an immunosuppressive drug. It's the same Trichoderma. This is generally found in the roots, around the roots of plants in the soil. This will not allow harmful microbes to gain entry into the plant. Thereby, it will prevent the plant from getting any diseases and infections. So, it can control plant pathogens from causing diseases in plants. Virus is also useful. There is a group of viruses called baculoviruses, more specifically a genus called nucleopolyhedrovirus is used extensively as a biocontrol agent. You know how viruses are extremely species specific, right? If they will generally parasitize only one type of species. Whatever viruses cause diseases and infections in us cannot cause disease in another species generally. So these baculoviruses generally target insects and other arthropods. Okay, so especially this genus, because they are very species specific and have narrow spectrum insecticidal application, they will not have any impact, negative impact on the ecosystem. If one nucleopolyhedrovirus targets only one species of insect, it will infect only that. The other insect species will not be harmed. Therefore, the ecosystem's biodiversity is not compromised. Okay, so that's baculoviruses. Last, we have microbes as biofertilizers. You know what fertilizers are? Fertilizers are chemicals that will enhance the nutrient quality of the soil. It will make the soil more fertile. Plants require nutrients to grow, which they absorb from the soil. If in the soil there is not sufficient nutrients available for its growth and development, we say that the soil is not very fertile. When it's not fertile, we add chemicals to increase its fertility. But again, adding these chemical fertilizers has the same disadvantages that chemical pesticides have. It can, make the plant products toxic. We could fall sick if we eat them. It will cause pollution of the soil and groundwater. These are some disadvantages of using chemical fertilizers. So what can we do instead? We can use living organisms as fertilizers. Generally, what is the soil deficient in? Usually, nitrogen will not be sufficient, or phosphorus or potassium. That is why they use NPK fertilizers, chemical fertilizers. There are microbes which are known to have the ability to fix atmospheric nitrogen. They can convert molecular nitrogen present in the atmosphere into absorbable forms, into forms that are absorbable by the plant. Of course, in our atmosphere, more than 70% is nitrogen, but plants cannot absorb that molecular nitrogen present in the air. They can only absorb it in the form of ammonia, nitrates, nitrites. There are microbes that can convert molecular nitrogen into these forms. So if you introduce those microbes into the crop field, into the soil, then nitrogen deficiency is taken care of. And there are certain fungi which form mycorrhizal association. They help in better absorption of phosphorus. By using these living organisms as fertilizers, all of those disadvantages can be taken care of. Generally, biofertilizers are bacteria, fungi and cyanobacteria. Bacteria may be symbiotically associated with the plant, or they may be free living. Example for a bacterium that is symbiotically associated is rhizobium. This is associated it has a mutual association with the roots of leguminous plants. Over there it will form small nodules. Within the nodules it will help in fixing nitrogen. In turn, it will get food from the plant. Okay, and then we have azospirillum and azotobacter. These are free living bacteria not specifically associated with any plant, and they also help in nitrogen fixation. Cyanobacteria, oscillatoria, anabena, nostoc, all of these are also helpful in fixing nitrogen. Generally, they are found in rice fields. They are used in rice fields for nitrogen fixation. Okay. Yeah, it serves as an important biofertilizer in paddy fields. It's also important because it adds more organic matter to the soil, which is again good for the quality of the soil. Finally, we have fungi. I told you that fungi forms mycorrhizal associations with the roots of higher plants like gymnosperms and angiosperms. There is one genus of fungus which specifically forms mycorrhizal associations, which is glomus. So what does this mycorrhizal association help in? Obviously, since fungi are filamentous in nature, it will increase the surface area available for absorption. It will help in the better absorption of phosphorus from the soil, and it also provides additional advantage. It will provide resistance against root-borne pathogens, increases the tolerance to salinity and drought where there is less water conditions, and overall increase in plant growth and development. So that is biofertilizers. Throughout this chapter, as you saw, all we discussed was which microbe is beneficial for what. All that we've discussed so far I've mentioned in the form of points over here, which you can go through and use to memorize and revise very frequently so that you do not forget anything. Alright, so that's about it. I hope you found today's lecture helpful. If you have doubts, please feel free to drop it in the comments. I will answer them as soon as I can. I'll see you in the next class with the chapter Organisms and Populations. Until then, take care of yourselves. Don't stop studying. Keep studying hard and enjoy the whole learning process. All the very best, and thank you.